Good afternoon. Oh, thank you. Welcome. I'm Barbara Schneider, president of Case Western Reserve University, and I'm so happy to see so many people in the audience. This is really a great day for Case Western Reserve. We have our alumni back and, of course, a great class of students on our campus, and we have a lot of people whose support makes lectures like this one possible. You first heard about Dr. Gary Landreth's amazing breakthrough in the potential treatment of Alzheimer's disease last February when it made national and indeed international news. His findings, published in the journal Science, showed that the use of a drug currently prescribed to treat cancer appears to reverse the symptoms of cognitive decline in mice with Alzheimer's disease. The results point to a significant potential that the medication involved, bexarotene, has to help the roughly 5.4 million Americans with this progressive brain disease. So powerful was the reaction to this news that I personally fielded requests from the families of patients with Alzheimer's disease who asked whether they could get their loved ones in the clinical trials. Just so you know, I have nothing to do with getting people into clinical trials. But it was a really powerful statement about the importance of this research. Intermediate studies will be required before that day, but I think Dr. Landreth's discovery is benefiting patients and their families in another way, and that's by providing hope, which is really important. Dr. Landreth actually holds the Archie and Ryuko co-chair in neuroscience, and the co's are with us today, and I want to recognize Archie and Ryuko co. Archie is a graduate of Case Western Reserve and a trustee, and he and Ryuko came to visit us today all the way from Singapore, and we are so glad that you are here. Please stand up and let us thank you. Gifts like theirs make it possible for us to recruit and retain outstanding faculty like Dr. Landreth. So to both of you, this is such an important gift, and I know that Dr. Landreth holds the co-chair with a tremendous amount of pride, and we are truly grateful. Research underway at our university is cause for much optimism, not just what you're going to hear about today, but other things as well. Recently, the School of Medicine and our dean, Dr. Pamela Davis, celebrated a landmark $64.6 million renewal grant from the National Institutes of Health for the Clinical and Translational Sciences Award, or as we call it, the CTSA. That was the single largest NIH award ever given in Northeast Ohio. And it supports a research alliance that includes Case Western Reserve, the Cleveland Clinic, Metro Health, University Hospitals, and the VA. The goal is to take discoveries from labs to patients and clinics and hospitals, or, as we like to say, from lab to life. And I mentioned Dr. Davis because leading that charge is our medical school, Dean Pamela Davis, who is the PI on the CTSA grant. That's principal investigator in English. Pam became dean of the Case Western Reserve University School of Medicine in September 2007. She holds the Arlene H. and Curtis F. Garvin Research Professorship. She is professor of pediatrics, physiology, and biophysics, as well as molecular biology and microbiology. She holds 8 patents. And under her leadership, the School of Medicine has refined the Western Reserve II curriculum, which emphasizes the themes of scholarship, professionalism, and leadership, and is taught in concert with the basic sciences. It also stresses the generation of original knowledge because, as Pam likes to explain, the physician best able to evaluate new advances in medicine is the one who has actually conducted research, and she really believes that. It is with great pride that I welcome to introduce the speaker for today, the Dean of Case Western Reserve University School of Medicine, Dr. Pamela Davis.
Welcome to our program this afternoon, and thank you, President Schneider. I love talking about this Alzheimer's discovery because it crystallizes so much of what we're trying to do at the medical school, to advance discoveries into cures and to educate the next generation of medical leaders. So Dr. Landreth, our professor, will be the first to point out that much of the research in his lab is made possible through the hands of outstanding students like Paige Cramer. Since the news of this study broke, Paige has graduated to become Dr. Cramer, and she's also landed a wonderful opportunity at Merck. So I thank you, Dr. Cramer, for coming back to your very first alumni reunion. During Dr. Landreth's 23 years of service at the School of Medicine, he has been a prolific cultivator of neuroscientists. Paige is actually one of 30 pre-docs, postdocs, and fellows that he has mentored. Gary came to us in 1989, following almost 10 years at the Medical University of South Carolina. He received his training first at the University of Kansas, then the University of Michigan, and postdoctoral work at Stanford University. And today, he is professor of neurosciences and neurology and director of the Alzheimer's Research Laboratory at the School of Medicine. Just last May, as President Schneider indicated, he became the Ryuko and Archie G. Koh Professor of Neurosciences. He holds 1 patent and 6 patent applications, has over 140 published papers, and is broadly respected across the neuroscience community. Now, after this lecture, I invite you to visit casemed.case.edu/AlzheimerCure, all one word, to learn more about their work. And now I will turn it over to Gary and Paige to let them tell you about these exciting times.
So I wanted to thank Dean Davis and President Sutter for that lovely introduction. And I am very grateful for the Koh's gift that allowed me to take this endowed professorship, which I greatly appreciate. Thank you. So what we're going to do today is we're going to talk a bit about Alzheimer's disease. Now, the way Paige and I are going to do this is that we're going to tell you about Alzheimer's disease and our work in this area for about 30 minutes. And then in the remaining 30 minutes or so, we're going to open it up for questions, and we'd be delighted to answer anything you want to know within limits. So what we're really going to focus on today is, I think, a story that holds some hope for Alzheimer's disease. Now, I would point out that there have been 28 failed clinical trials for Alzheimer's disease, and that provides a setting for our work. So just as a point of reference, how many people in this room know someone or have interacted with someone with Alzheimer's disease? I think that's almost everyone in the room. So, you know, it's been 100 years since Alois Alzheimer saw the first patient, Auguste D., who came into his clinic. She was 51 years old. She suffered from profound memory problems and a disordered thinking. On her death, he examined her brain and for the first time recognized the canonical pathological hallmarks of the disease, which we now recognize as Alzheimer's disease. This was, until relatively recently, thought to be a rare disease. When I trained as a graduate student in the early 70s, we rarely saw a person with Alzheimer's disease, and sometimes it was called senile dementia. And so in the modern age, we recognize these are all the same things, and this has become an increasing problem for us as a society. So I don't think I need to tell you that Alzheimer's disease really represents the dysfunction of higher order information processing and recall. Indeed, it is the essence of what we are as human beings. This is the most common form of dementia. Now, there are several kinds of dementia, but about 85% of all the dementia in the community is, in fact, Alzheimer's disease. There are no effective treatments for the disease. We'll come back to that in a second. You know, this is the largest unmet need in medicine. This is the disease of the 21st century. What we have available for now, if you get a diagnosis of Alzheimer's disease, is a collection of drugs which fundamentally do not work. There are very limited options therapeutically for drugs that modestly affect the efficiency of the diseased brain. These are symptomatic treatments. They work in about 1 in 5 people for 6 to 9 months. This does not affect the course of the disease. And so that's currently the state of play for our treatment options in Alzheimer's disease. So I think the most common, I think we all sort of fundamentally understand what Alzheimer's is about, but people end up in the doctor's offices primarily because they suffer memory loss, and it's a very specific type of memory loss, the ability to form short-term memories. Long-term memories tend to be intact even until late in the disease, but it's your capacity to remember where you put your car keys, where you parked your car, telephone numbers, all the functions of very short-term memory formation, which occurs in a structure called the hippocampus. These patients have problems with spatial perception. I think it's, one of the common things is people get lost because they lose the capacity to place themselves in space. Moreover, as the disease progresses, there are losses in judgment, or what's been termed executive function. This is the capacity to problem solve and to exhibit good judgment as they have in their as young people. There is a panorama of behavioral changes which accompany the disease. There's problems with attention. Depression is a very serious problem, particularly in the early stages of the disease, as the individual sort of recognizes what's happening and what's down the road. In later stages of the disease, aggression is a very common form of behavioral disorder. And then there's apathy. So this constellation of symptoms represent what we now recognize as clinical Alzheimer's disease. So this is the bad news. There is an epidemic. We are in the midst of an epidemic of Alzheimer's disease. Today in the United States, there are 5.3 million individuals with Alzheimer's disease. That number is going to triple by 2050. This is not just a function, this is not just the United States. Every industrialized country in the world has this same trajectory for Alzheimer's disease. And the reason for this is, in fact, because we're all living longer. This figure is actually rather stunning, because what it says is if you're age 65, about 2% of that cohort have Alzheimer's disease. As you move into your 70s, the number works out to about 15% or so. But the really dramatic thing, and that segment of the population which is growing most rapidly, are those individuals over the age of 85. And roughly half of all individuals over the age of 85 have Alzheimer's disease. I mean, I think that's an amazing statistic. But this is the real problem. We've been having this discussion as a nation about the cost of health care. This disease alone has the capacity to break the medical system in this country. Today, it costs roughly $200 billion to care for the 5.3 million individuals with the disease. And that's because it costs maybe between $20,000 and $40,000 a year per patient to care for those people, largely due to the costs of custodial care. By 2050, that number will be $1.1 trillion for Alzheimer's disease alone. The cumulative total to the economy over the interval between now and then is about $20 trillion. Again, the current discussion about the costs of Medicare and healthcare in this country will pale into insignificance unless we solve this problem. Now, the reason why we are in the midst of an epidemic is we're all healthier and we're living longer. And if you look at the cause of death, what you'll see is currently Alzheimer's is now the 6th most common cause of death in the United States. But that's because we've largely fixed a lot of the problems in biomedical science. Indeed, the fact that we're getting Alzheimer's disease represents, in an odd way, the success of modern medicine. Because if you look here in this interval, what you find is HIV deaths are down by about 25%, heart disease is down another 13%, cancers are all down, but Alzheimer's disease increased by 66% in that interval. I mean, that's a shocking statistic. And I don't think it's unrelated to this slide. If you look where we put our dollars from the National Institutes of Health into medical research, what you see, we have dropped a lot of money into cancer, heart disease, and HIV. And what I just showed you is that the rates of death for those diseases have fallen rather dramatically. So that as you invest in biomedical research, we solve these problems and we live longer due to those specific diseases. Now, the important factoid I want you to pay attention to is that we put under $500 million into Alzheimer's disease research. And I just showed you the net cost of this disease to this society. This disparity is going to underlie the entire discussion about whether we can fix this disease in this society in time to prevent a both medical and economic catastrophe. So, what causes Alzheimer's disease? Well, for about 5% of us, you get your Alzheimer's disease because of genetic mutations. So this is that there are 3 genes which can be mutated, and you, with 100% fidelity, will get Alzheimer's disease. But it's a very modest number. These people and these families get their disease in their 40s, 50s, and early 60s. The rate at which the disease progresses is very rapid. And these people have been studied and told us important things about how the disease occurs. But for 95% of us, 95% of cases, there's a thing called late onset Alzheimer's disease or sporadic Alzheimer's disease, and that occurs in individuals largely over the age of 65. I think it is a testament to our ignorance is that we do not know what causes those 95% of Alzheimer's cases, although Paige will have something to say about that in a second. So what Alzheimer's recognized when he looked at the brain of that first patient in Alzheimer's is that the brain is shrunken. And there is a dramatic loss of neurons, so that is the elements which communicate between each other and which are essential for brain function, are lost. And specifically, what is lost is those areas of the brain that are focused on the formation of memories. This is called the hippocampus. And also areas in the temporal lobe which are involved with language, which are the first 2 things to go, which suffer a disproportionate and the earliest loss. And indeed, I think that this is a rather graphic demonstration of the devastating nature of the disease in humans. So, what underlies the extensive neuronal death and the pathology in the disease? So the first thing that appears as you look in individuals that are at risk for the disease is the formation of what's been termed amyloid plaques. That form and are deposited in the brain. We'll talk about that more in a second. But also the neurons, that is the central elements for communication in the brain, have been disabled and are dystrophic. That is, they're diseased, as evidenced by what are called neurofibrillary tangles. And that's just a sign that things are going very badly for this cell, and it's on its way out. The constellation of these 2 pathologies contribute to the inability of neurons to communicate with one another. And this is what underlies the loss of cognition and memory. Eventually, as the disease progresses, the neurons themselves die, and that's when you get into the more advanced, profound cognitive deficits we see in advanced stages of the disease. We now believe, based on both animal studies and in humans, that what happens first is the deposition of amyloid within the brain. And what that does is causes a cascade of events, which ultimately impacts the neurons themselves. So this is an amyloid-driven neuropathy. Now, What I want to focus on mostly is the formation of amyloid plaques in the brain. So in advanced cases of the disease, which you can see here, is up to 30% of the volume of the brain can be occupied by these amyloid plaques, and that's correlated with the loss of neurons in the brains of these individuals. Now, when I say amyloid plaques, what I really mean is that these are waxy deposits that appear and are formed within the brain itself. And the primary constituent of these plaques is a peptide called beta amyloid. Now, as you're listening to me, we're all producing prodigious amounts of beta amyloid as a normal consequence of brain function. That is, as your neurons fire, they generate this amyloid, and we actually don't know why. But what we do know is that we're all producing prodigious amounts all the time. And it's secreted from neurons as these little bitty things are called monomers. And so the problem is that these things are rather sticky. And they stick to one another to form what are called oligomers. And these go on to form fibrils, and the fibrils go on to form plaques. In the brain, in the Alzheimer brain, what happens is that these small peptides gradually accumulate and are not cleared from the brain. So the more amyloid peptide you have in the brain, the more fibrils you get, the more plaques you get. So if you think about this as a potential therapy for the disease, if you could attack this process, you might have some impact on disease progression. And many of the currently investigated therapeutics for the disease focus on this cascade. So we've really made some important advances in the last decade or so. And in fact, we can diagnose Alzheimer's disease with almost 100% certainty. We do this through a use of rather sophisticated neuropsychological instruments where we can test memory and cognition and judgment. Furthermore, there's a thing called biomarkers, which means they can go in and take a tap from your spinal cord and collect spinal fluid and determine whether you have proteins related to these neurofibrillary tangles or the plaques. Those are called biomarkers. But perhaps the most important advance is the use of new imaging technologies. And you may have heard about this in the last few weeks, because there's been a brand new imaging agent called amyloid, which was just approved by the FDA just about a month ago. Now, the important thing about this is that you can go into the doctor's, into a hospital setting. They can give you an injection of amyloid. And what it is, it's a dye that sticks to amyloid plaques. Then they put the patient into the scanner, and then they can determine if and where amyloid plaques appear. And this is shown rather graphically here. So this is a cognitively normal individual. This is an age-matched person with Alzheimer's disease. And what you see is the Alzheimer's patient has lots of plaques in their head, and the dye binds to those plaques, and then it can be imaged in the machine. So what that says is that we can non-invasively determine whether you have, or the patient has, amyloid deposition, which is the prodromal state to Alzheimer's disease. This is a really important technical advance that has been achieved recently. And what this allows us to do is not only to diagnose the disease, but we'll be able to measure whether the drugs we design and employ to treat the disease are efficacious. Right now, we have to wait till the patient's dead and do an autopsy to determine whether any effective treatment is worth. So I can't overemphasize the importance of the money the NIH has invested in these types of technologies. And it will power drug development as we go forward. When we got these new imaging tools, there were a group of clinicians who went back and did large studies of cognitively normal people in their 60s and 50s. And so the new news, and in fact the shocking news, is that most individuals show the signs of Alzheimer's disease in their brain up to a decade and a half before they show up in the doctor's office complaining of memory impairments. I think this fundamentally changes how we think about treating the disease, because I think that means that people at risk for the disease, which I think is all of us, may have to engage therapies in our 50s and 60s. And if you have a really dramatic risk factor, you have a family history or something, maybe we'll treat these people in their 40s. This changes everything, because this is the curve of amyloid deposition in the brain as imaged by this new imaging agent. And you see that these cognitive neural people can have actually quite a lot of amyloid in the brain before they become cognitively impaired, which is shown by the green curve. And again, this is 1 to 2 decades. I mean, it's a shocking finding, and I think we all need to attend to that. So, there have been important new advances in the last year or two, largely out of a group in St. Louis. Now, as we thought about this, we thought, okay, the reason why amyloid accumulates in the brain is we're making too much. And that was a thought that drove a lot of thinking in the disease. There were new studies that were published about 2 years ago that said that's not it at all. And I'm going to read this to you. Is that it turns out with most people with Alzheimer's seem to make perfectly normal amounts of amyloid. They just can't get rid of it. Like an overflowing sink caused by a clogged drain instead of a faucet that does not turn off. So what this says is that the reason most of us will get our Alzheimer's disease is because we cannot appropriately get rid of or normally clear the amyloid that is formed as a consequence of normal brain function. So, my lab, over the last decade, has been focused on trying to understand how the brain rids itself of these amyloid peptides. So the neurons produce these amyloid peptides, and in cognitively normal individuals, in young individuals, that these are effectively clear. That is, that they're cut up into small bits and passed out into the circulation and cause no harm. And when you can do that efficiently, you have normal cognition. Now, the difficulty arises is that as we age, there are age-related impairments in this clearance mechanism. Now, I think we all recognize that, aging's not so great, right? And we all have different systems go wrong at different rates in different people, right? Same thing in the brain. Some of us are going to be very efficient in clearing your amyloid and until late age. I mean, like Bob Hope was 100, and he still had all of his marbles. But some of us won't. And this is just a consequence in the brain of aging. The problem is, as we become less efficient in clearing amyloid out of the brain, it accumulates. And these small peptides, these small forms of amyloid, basically poison the communication between neurons, that they interfere with synaptic transmission in the brain. And this is what underlies the cognitive impairments in the disease. As time goes on, what happens is these peptides accumulate, they form fibrils, and they're deposited into plaques. And then this contributes to the neuronal loss in the disease. So in combination, it is the failure to effectively clear amyloid from the brain which underlies all the subsequent pathologies. So we thought, let's go try and find drugs and the biological mechanisms that underlie clearance and target those with therapeutics. Now, I've had the great good fortune to work with an extraordinarily talented group of young scientists, which basically do all the work in my lab. So in 2007, I had an extraordinary graduate student show up and wanted to work on this problem. And I think she'll tell you about what she did.
Thanks, Gary. So let me fast forward. So fast forward to 2008. So I've been in the lab about a year. And among other graduate students in the lab, we'd figured out an ability of a protein known as ApoE, or apolipoprotein E. And what ApoE does, it actually acts to facilitate clearance of that soluble amyloid beta peptide out of the brain. So you can think of this protein as a garbage disposal. So the more garbage disposal you have in your brain, the faster this amyloid beta peptide is cleared out of the brain. And what that is doing then is preventing the synaptic, preventing the synaptic deficits. Let's see, there we go. Can you see? Okay, the synaptic deficits. It's also preventing or reducing the number of amyloid beta plaques within the brain, and thus you have a healthy brain. So then fast forward about a year, 2009, we were interested in looking for drugs that do exactly this, increase that garbage disposal within the brain. So bexarotene is the drug that we have found. This drug does increase ApoE within the brain, and fortunately for us, this drug is FDA approved. This is also really exciting because we believe it will actually speed its development for the treatment of Alzheimer's disease, something that we're all interested in doing. And fortunately for us, because it is FDA approved, it's passed through a lot of the hurdles and hoops. It has a good safety and side effect profile. So again, this drug, bexarotene or targretin, increases ApoE within your brain. And what ApoE is going to do, it's going to shuttle out the soluble forms of amyloid beta out of your brain. It's going to prevent the synaptic deficits or those neuron-to-neuron communication failures. It's also going to reduce the amount of plaques within your brain, thus preventing the cognitive impairment that's associated with the disease and promoting a healthy brain. Fortunately for us in the laboratory, we have animal models of the disease. And as Gary mentioned, there are about 5% of the cases of Alzheimer's disease are accounted for by genetic mutations. And previous laboratories have actually taken those genes that are mutated in people with Alzheimer's disease. They've inserted those mutated genes into the mouse genome. And then, fortunately for us, these mice now exhibit some of the symptoms of Alzheimer's disease that include cognition and memory impairments, as well as the amyloid beta plaque deposition. So there's just a couple of true research slides in here. So one of the first questions we wanted to look at is whether bexarotene could clear that soluble amyloid beta out of the brain. So we use a technique known as in vivo microdialysis, where we have awake, freely moving mice. We insert a probe into their brain, into the memory center or the hippocampus of the brain. We can then measure the fluid that surrounds the brain and look at amyloid beta plaque or amyloid beta levels. So you can see here in the open circles are just your vehicle or your placebo-treated animals. And on the black boxes are the bexarotene-treated mice. So as you can see in our time frame here, we're looking at a pretty short time frame. And after just one dose of bexarotene, shown here by the downward arrow, you can see there's about a 25 to 30% reduction in soluble forms of amyloid beta. Now, something that's really important and really exciting for all of us is that actually just one dose of this drug, one dose of bexarotene, can actually reduce levels of soluble forms of amyloid beta for up to 72 hours. So that is astounding for any drug developer out there. So the next question we wanted to look at, after we now realize that bexarotene can clear or can facilitate the clearance of soluble species of amyloid beta, we wanted to look at the plaques, the pathological features of Alzheimer's disease. So shown here on the top panel, this is a vehicle-treated animal, just a water-treated animal. The blue represents nuclei within the brain, so all of the cells within the brain. And if you can see the red is actually a stain for amyloid beta. Now, if you just do a direct comparison of the cortex of vehicle compared to the cortex of the vehicle or the bexarotene-treated animals for 14 days, there's a dramatic reduction in the amount of red stain there. Now, what that tells us is that there's a reduction in the plaque area of these animals after 14 days of bexarotene treatment. And that was also recapitulated in the hippocampus, or the memory center of the animals. So this is something that is incredibly exciting to us because this time frame is rapid. We're seeing dramatic reductions in 72 hours. So very few other researchers out there, in fact, I can't name any, to be frank, have seen such dramatic effects within a short amount of time. So now that we have reductions in soluble forms of amyloid beta, we have reductions in the actual plaques, those waxy substances, we wanted to look at the cognition and memory of these animals. And as I mentioned before, fortunately they have these deficits. One of the easiest tests to look at in mice is their nesting ability. This is an associative behavior. So if you look at the wild type, the top left-hand side, you take a wild type mouse or your garden variety mouse, you give it pieces of pressed paper or pressed cotton. Typically these mice will chew it up, spit it out, make a burrow on the side or on the corner of their home cage. However, if you provide pieces of pressed paper or pressed cotton to the Alzheimer's disease mouse, they've forgotten this associative behavior. They have no idea what to do with the pieces of paper. Fortunately for us, if you treat these animals for 3 days with bexarotene, these animals now begin to make a nest. They now remember this associative behavior and are making their nest building. Though it may not look exactly like a wild-type mouse, they are, in fact, remembering this associative nesting behavior. So as Gary mentioned before, people with Alzheimer's disease have spatial misperceptions. They get lost. They don't know where home is. Fortunately for us, the same concept applies in mice with Alzheimer's disease. We have a test that's known as the Morris water maze. And in this test, we put mice in a pool of opaque water. Mice don't like to swim. So we also have a hidden platform within this pool. So over the course of 7 days, these mice learn to orient themselves, spatially orient themselves with markers that are in the room, like this red circle, blue star. So they can orient themselves within the room. And on the last day of training, the last day of testing, we take out the hidden platform, and we look to see how long did the mice spend in the correct quadrant where the hidden platform was. Oops. So your normal mouse spends about 30 seconds of the amount of time, it's at a minute test, in the correct quadrant, meaning it's swimming back and forth into that little quadrant, looking for that hidden platform so it can stop swimming. The AD mouse, or the Alzheimer's mouse, spends about a quarter of its time in every single quadrant, or about 15 seconds in the correct quadrant. So it doesn't remember where it is, it doesn't remember where the hidden platform is. But importantly, and more excitingly for us, is the Alzheimer's disease mouse that was treated with bexarotene now spends a significantly more amount of time in the correct quadrant, which indicates to us that it is now remembering where the platform is and has improved spatial memory. So hopefully what I've shown you just a little bit about today is that this drug, bexarotene, can rapidly reverse these Alzheimer's disease-related pathologies in the mouse models that we're using. And again, this is a mouse model. You have to keep this in mind. What it is doing, it is increasing ApoE in the brain, that's something I actually didn't show you, but take it from me, it really does increase ApoE within the brain. It then facilitates the clearance of soluble forms of amyloid beta out of the brain. It reduces soluble levels then. It reduces the amount of plaques within the brain, and it also improves their behavior, meaning the synaptic deficits are gone. There's now perfect communication between the neurons. So where are we going and what have we done? So this is a proof of principle study in mice. The next steps are really to figure out how much drug do we need to give mice? How often do we need to give this drug? We want to make sure that we're not over-medicating, and we also want to make sure we're not under-medicating the animal models we're using. We want to be as optimized and efficacious as possible. These studies are going on right now in Gary Landreth's lab. More excitingly, and I think what everyone is really interested in, is initiating human clinical trials. Now, everything is kind of on the move to begin these trials within the year, and I think I'll tell you just a little bit more about it, but that is our ultimate goal, is to move this bench to bedside, move this information from the bench to the bedside. So what do we have? We have translation into clinical trials. We have this phase 1b clinical trial. It's referred to as the proof of mechanism clinical biomarker trial of bexarotene in humans. It will be a randomized, placebo-controlled study looking at both A-beta as well as ApoE metabolism within the central nervous system. So this study will be comprised of 12 individuals. They will be cognitively normal, younger patients, 6 of which will be given the drug, 6 of which will be given placebo. We will look to make sure that really this drug that's working in mice, we want to make sure it works the same way in humans. This is expected to initiate within the next couple of months, and actually even more excitingly, is that this trial will only take 4 months. So in terms of clinical trials, time-wise, this is very rapid. We're working with fantastic individuals that have incredible techniques that we can really speed the development. So this, of course, would not have been done without the work here at Case Western Reserve University, specifically in the School of Medicine. Fortunately for us, they have launched a website, as Dean Davis mentioned earlier. It's casemed.case.edu/Alzheimer's Cure. This is a way for you to stay interactive with us, the researchers that are at the bench. You can track our progress. You can see what we're doing. You can ask us questions. This is also a way for you to share with your friends what's going on in Case Western Reserve University. It's also a way for you to make a gift. Obviously, all of the work that we have been doing here has been supported by philanthropy, and we want to get to the next stage, which of course requires more philanthropy. So the other interesting thing here is that I think on everyone's seat, there was a piece of paper and a pen. If you'd like to stay connected with us, we would love that. Feel free to fill out the forms, and I think there should be some people in the back that would be happy to take them. And that is just a way for us to say, one, thank you so much. All this work would not have been done without your support. And also, please stay in touch. We are pretty anxious and excited to see what comes next. So we'll take any of your questions at this point.
In a recent medical school alumni bulletin, the journalist who wrote up the article about what's going on in your lab said that the initial trials are going to be with normal patients.
Correct.
But it didn't sound like that the way you described it, but of course it's very sketchy. And if you would say, hopefully, a good deal more about the nature of the subjects and,
sure.
And what, to what extent, what your protocol is with respect to going from perhaps relatively normal to a more diseased population?
Sure. So initially, what will happen is we'll have 12 cognitively normal individuals. They'll be young individuals, probably in their 30s. They will be given drug. Half of them will be given drug, half of them will not. Hopefully at that point we'll see a rise in ApoE within the central nervous system. We'll also see a fall of A-beta or a change in A-beta levels within the central nervous system. That will be a validation for us that this drug is working in people as we've shown it does in mice. At that point, we have, in terms of the trial design for the phase 2 and phase 3, those have not been designed at this point. We're waiting for the phase 1 information to come through. Gary may have more.
So if I could say, so this is a cancer drug.
Yeah.
It has never been examined, its actions in the brain have never before been examined. There have been 28 failed phase 3 clinical trials because of poor trial design and poor reagent. We're not going to make that mistake. We're going to do this methodically. Also, you know, what we're worried about and why we're going to normal individuals first is that if this drug is as good in mice, or good in humans as it is in mice, we're removing plaque from the brain. And I just showed you a slide with the brains of Alzheimer's patients loaded with plaque. If we remove those plaque, what happens? You end up with voids in the brain. This has never been done before. I think there are serious safety considerations which need to be considered before we go into people with advanced disease. We can't make an error. We can't have an adverse event. We need to do this slowly and methodically. So the other piece of this, we have partnered with a company called C2N Diagnostics in St. Louis. We are using state-of-the-art space-age technologies to evaluate amyloid metabolism and ApoE metabolism in the brain. This has never been done before. So I think the pattern for drug development in Alzheimer's disease subsequently is going to be written around the development of this drug. John.
Is the structure of alpha-beta amyloid in the brain similar to the structure of the amyloid that's deposited in the islets of Langerhans in people with type 2 diabetes?
So what they share is what's been termed beta-pleated sheets. So the reason why these peptides are sticky is that they have basically sort of a ribbon structure, and the ribbons stick together. So the fundamental structure of these proteins and their capacity to form these amyloid plaques is the same, but the individual proteins are quite different.
Do you think the drug that you're developing might reduce the amount of amyloid in the islets?
So the answer to that is no. Because the reason why the plaques go away is basically what we do is turn on the capacity of macrophages to gobble up or phagocytose this in the brain. The biology in pancreas and elsewhere is quite different. I mean, it could be, but that isn't our expectation up front.
I have two questions. The first one is, what are the ethical implications of administering this drug to people with normal cognitive function? Is there a possibility of improving memory to a point that would be considered superhuman?
Yeah.
And second of all, sorry, when you remove amyloid beta from the brain, does it have a natural function And are there functional effects to taking it away?
No. Okay, those are two excellent questions. One is this drug has been in clinical usage for T-cell lymphoma for over 10 years. There have been no either positive or negative effects associated with its use in that population. There have been thousands of people who are on this drug for their cancer. Last month, in the New England Journal of Medicine, there was an article by bioethicists discussing the appropriateness of using this drug off-label, because you can go to your physician and he can write you a prescription for this drug and go down to the drugstore and you can take it. Now the question is that something you really want to do? And what the bioethicists point out is not really. This is not an appropriate thing to do for at least for a physician until the underlying biology is sorted out and we know that it's safe. You want to answer that?
I was going to say, to answer your other question in terms of amyloid beta in the brain, if you take it out or if you overexpress it or underexpress it, there's not really a change, at least in the mice. So in terms of removing the amyloid beta, the plaques themselves, we've not seen any negatives. It's only been positive. And in terms of just animal models themselves, if you overexpress or underexpress amyloid beta, there's really no difference.
That is, when this drug is used for cancer, what are the side effects? How is it excreted? Do you get renal failure possibly? And the second question is, in patients with this cancer who have Alzheimer's disease, is there anecdotal observations?
So the first question in terms of it's given at a very high dose, and the side effect profiles of those patients that are on it for the cancer indication are hypercholesterolemia and hyperdyslipidemia. Those are also managed by other medications, genofibrates. So in that sense, it's safe. And then the second question, in terms of, I'm sorry, can you say your second question again?
Well, part of the first question was then, how is it, what does the body do with it? And then the actual second question was, are there anecdotal observations?
So in terms of the excretion, it's broken down into about 3 or 4 different metabolites and are excreted. The anecdotal evidence, unfortunately, is such that patients who do have cutaneous T-cell lymphoma, unfortunately, this is a 3rd and 4th tier medication for those patients. Also, the patients that do have cutaneous T-cell lymphoma are much younger. They're not of the age to be developing Alzheimer's disease, and as it is a 3rd tier, or 2nd or 3rd tier drug, there's not any anecdotal evidence. That being said, we have interacted with a few dermatologists at Case Western Reserve University, and I think that's something that we will definitely be interested in looking forward to.
Why are some parts of the brain spared from the amyloid deposits?
Oh, $64,000 question. So you're not the first person to ask that question. So that's one of the great enigmas. I mean, why is it the hippocampus, and why is it certain sections of the cortex but not other parts of the brain?
Yeah.
So there have been some rather interesting new pieces of data. So metabolically, those are the most active parts of the brain. So you're generating more amyloid there just as a consequence of brain activity. The new news is that both the amyloid and this neurofibrillary tangle, it's called protein called tau, are secreted into the extracellular space and are taken up by the neurons to which they connect. And it's been shown that what happens is this pathology is propagated through synapses, through neural networks. So hippocampus is affected first. It communicates with cortex and then is disseminated to other parts of the brain. Now that's a bit controversial, but that's how we're thinking about it now. Yes.
Yes. What is your opinion on reports that the B vitamin niacinamide, the non-flush form of niacin, dissolves amyloid?
So I'm not, I'm not familiar with that literature in the periphery, but as far as I know, those reports have not sort of reverberated into the CNS. Now, what we've talked about, what Paige talked about, is elevating ApoE, and so you'll know this as high-density lipoproteins, or HDLs, and niacin performs that function in the periphery. It's never been shown, to my knowledge, to change brain HDL levels.
Thank you.
How often would you expect to administer the drug in order to continue the effects?
So that's something we're working on right now. At this point, one, we're looking at a far lower dosage than the FDA-approved dose. We're also looking at maybe an every other day, every third day treatment in mice. That's something that we are actively pursuing in the laboratory now, but we don't have hard, fast data on that yet.
How did you go about discovering that this drug would be effective?
No. Okay, well, you know, so it took us about 10 years, and if we were smarter, it had gone faster. So my laboratory has worked on the machinery, the really basic biochemical machinery to elevate ApoE for a decade. So we know something about how that machinery works. And so we were provoked to think about this. It's called a retinoid X receptor agonist, bexarotene. And there were a zillion reasons why this should not have worked. This would never have gotten past an NIH review panel because we predicted that the side effect profiles would be much worse. And science is serendipity. And God bless, this drug worked with modest side effects in the brain. I mean, to a degree, we just got lucky. Like there was a little bit of brain work involved there, but oh yeah. So we've got a we've got a nice collection of drugs we're after, and you know the truth is, since February there are a lot of other people working on this problem now too. So I think we will not, if this underlying mechanism is correct and we got it right, there will be no lack of drugs like bexarotene that are going to be in clinical development in relatively short term.
I wanted to ask about the link between Alzheimer's disease and primary light chain amyloidosis. I have light chain amyloidosis. I have amyloid deposits in my duodenum. I went through chemotherapy with melphalan in 2000 at Boston University School of Medicine, and stem cell transplant. I've been in remission since 2000. My question is there a good possibility that I might still be vulnerable for Alzheimer's disease because my mother had it, and she died after about 10 years. She died about 85. Yeah.
So I think these are two unrelated things. I think that the disease, the peripheral disease, is unrelated to the Alzheimer's pathologies, and you're at risk for Alzheimer's disease because you have a first-degree relative who had Alzheimer's disease. But I think that's completely unrelated to the other disorder.
Are you finding any significance for what you're going to be doing in terms of the different ApoE subtypes, 2, 3, and 4?
That's a fantastic question. So for those people who do not know, people have, so the question is, so ApoE comes in three different flavors in humans. So you have ApoE2, ApoE3, and ApoE4. There are risk factors associated with each of these isoforms, meaning that if you have one copy of ApoE2 from your mother and one copy of ApoE2 from your father, you are at a very reduced risk for developing Alzheimer's disease. On the counter to that, if you have one copy of ApoE4, you're about 3 to 4 times more likely to develop the disease. And if you are unfortunate enough to have one copy of ApoE4 from your mother and one copy of ApoE4 from your father, you're about 12 times more likely to develop the disease. ApoE3, however, is the predominant form of ApoE that people have. About 77% of people have at least one copy of ApoE3. And in terms of the studies here, the mouse ApoE is very close to an ApoE4 mouse, or an ApoE4. However, that is not to say that human ApoE4 is resembling mouse ApoE. That's something that there are now new models of Alzheimer's disease that actually incorporate human ApoE2, 3, and 4 isoforms. And actually, we have a collaborator in Pittsburgh who is looking at bexarotene treatment in APOE2, APOE3, and APOE4 mice. And I think she has seen behavioral improvements in
mice bearing the E3 allele,
yeah.
They see exactly the same effects that we see in our mice. The question about APOE4 has yet to be answered.
Yeah.
It's controversial. Some people think that increasing APOE4 will be a bad thing in the brain. We need to examine that question in mice first. And this is another reason why you should not take this drug off label. So we're working as fast as we can. Don't know.
Any link to exercise and staving off the onset of Alzheimer's?
So I would say what is good for your heart is good for your brain. So anything that is beneficial cardiovascularly is also going to be beneficially to your brain. There are a lot of studies in terms of exercise and Alzheimer's disease, and those I think are still ongoing. Obviously, what's good for your heart is good for your brain.
I fully understand the rationale of detailed studies doing it right. Believe that. But based on my neighbor and what he's gone through, I can tell you, you could have thousands of volunteers right now who would not care if they died, all right, if it would move this ahead, all right.
So I can tell you, Paige and I published this paper on the 8th of February. I have yet to be able to answer my telephone. Yesterday, 2 calls, where we have had hundreds of emails, phone calls, letters. The desperation in the community is profound. I can't tell you how moving this is. And I can tell you that there are a lot of people on this drug off-label because there are no other options. I mean, this is a devastating disease for which there are no options. I fully understand that. And we've got to work faster, we've got to work smarter.
Okay.
Well, if that's the last, I want to thank you all for coming this afternoon. It was a pleasure to talk to you.
Thank you so much for coming.